Coming up on Dr. Kiki's Science Hour, we're talking about retroviruses with Abbie Smith from Science Blogs. That's up next on Dr. Kiki's Science Hour.
Netcasts you love.
From people you trust.
This is Twit. Bandwidth for Dr. Kiki's Science Hour is provided by Cachefly at CACHEFLY.com.
This is Dr. Kiki's Science Hour with Dr. Kiki, episode number 70, recorded on Thursday, October 28, 2010. We're going retro. Welcome to Dr. Kiki's Science Hour. I'm Dr. Kiki, and this is episode 70. Can't believe it, can you? It's a nice round number, but it's kind of big. I can't believe I've been doing the show this long. But there's always more science to talk about, and we've got some exciting, interesting, it's kind of a mystery of science show for us today that I hope we can really delve into. Our guest today is Abbie Smith. She has a blog on Science Blogs called ERV, which is short for endogenous retroviruses. So in effect, we're going retro. We're talking about those viruses that dig themselves into our genome and kind of stay there. And what do they do? What are they for? What's going on with these viruses? Hopefully Abbie can shed some light on that. Also, I'd love to get into some topics of she's a great skeptical blogger and has had some great conversations online with lots of the top skeptic bloggers, including Pharyngula and among others. So we'll hopefully have a little bit of fun with that as well. Without any further ado, as we start to get deep and dirty into the subject of retroviruses, Abbie is a graduate student at the University of Oklahoma, and she studies the evolution of viruses, especially retroviruses, and as she says on her blog, how that evolution impacts our health. So without any further ado, I'd love to welcome Abbie to the show. Thank you for joining me.
Thanks for asking me.
You're welcome. So let's get this started with just the question of what, can you explain the retrovirus?
Sure.
What is it?
So most organisms, we carry our genome as DNA, right? And that's sort of just a storage mechanism. It gets converted into RNA, which then it's the instructions for how to make proteins. Well, retroviruses do things a little differently. They store their genome as RNA. It gets turned into DNA. It sneaks into our genome and convinces our cells that, oh, this is just a normal cellular gene, so let's go. And it tricks our cells into making these viral particles that but often go to infect more cells. So what happens with endogenous retroviruses is that instead of infecting a cell that it means to infect to create more progeny variants, a retrovirus might accidentally infect a sperm or an egg, and it will permanently become part of that organism if it goes on to be fertilized or to fertilize an egg and grow up into an organism.
Wow. So how do we know how a virus can get into a sperm or an egg cell? I mean, the germ line is like, it seems like it's protected. I mean,
yeah.
I can understand, okay, you can get into the lungs, you can get into anything that kind of associates with the outside environment, but it seems like getting into the actual sex cells, the things that carry on that information, would be really difficult.
It is, and it is a complete accident. A lot of the times with viruses, things happen on accident all the time because you're dealing with billions and billions and billions of particles that have infected billions and billions of people or organisms over the history of evolutionary history of this planet. So highly improbable events have actually happened numerous times over the evolutionary time scale.
I love the it's improbable, but it happens.
Yes. Well, yeah, highly improbable events can happen all the time when you're dealing with billions and billions of years.
Yeah. Looking at your blog, you do explain in a number of places that a lot of these ERVs, these ERV, endogenous retroviruses, they're in there in our DNA, but they're considered junk DNA. Do they, I mean, a lot of times, do they just not do anything? They just hang out?
Yes, and that is a very good thing. So let's say you have a very young endogenous retrovirus, like one organism, it's the very first organism to ever have this endogenous retrovirus. First of all, it could kill that embryo on impact. But your genome has evolved different mechanisms over our evolutionary history to silence these endogenous retroviruses when they still are active or have the potential to be active, and that's epigenetics. I hate to bring up that word without totally defining it, but it's a way to wrap up your DNA really tight so it doesn't make RNA to go on to make those proteins. So if there's a retrovirus in your cell and you can wrap up that retroviral DNA super, super tight, then it's effectively dead. It can't go on to make any proteins. So what was the question? I got sidetracked talking about epigenetics.
No, that's okay. So
the junk.
Yeah, the
that's
junk.
Right. Okay, so if you wrap it up really tight and it can't, directly kill that organism. I mean, we know what happens when retroviruses infect organisms. They get cancer or they get immunodeficiencies and can be killed by something else. I mean, they aren't evolutionary advantageous in any way when they're retroviruses. But what happens is when you can wrap them up, keep them silent, your genome doesn't really have a way to kick them out. You can't get rid of them. But what happens is they accumulate mutations over time because there's no advantage to having them and there's no disadvantage to mutating their functionality. Right. And if anything, it's kind of an advantage to make them non-functional. So if your epigenetics gets knocked off balance, you aren't going to accidentally start making these proteins. So yes, you have hundreds of thousands of endogenous retroviral proteins in your genome, and almost all of them have stop codons or nonsense mutations or huge deletions in them, so they aren't functional anymore. This is a very, very, very good thing.
Yeah, it would be a very good thing. So you get this genetic information from a virus into your genome. Through epigenetic functions, we're able to block them and keep them from being expressed and keep them from doing anything. Do we know about any examples of retroviruses suddenly becoming awakened? I mean, I've read numerous stories over the last several years. There's some French researchers working on retroviruses that they've isolated from either a human DNA or bacterial DNA that they've been trying to sequence, take out of the actual organismal DNA, sequence it on its own, and then replicate it and try and get that virus to actually live in a dish or wherever it is in a laboratory. Is this a danger?
Okay, so, I said you had hundreds and hundreds of thousands of these endogenous retroviruses in your genome. Well, some of them are kind of related to one another. So let's say you have 20 copies of this retroviral family in your genome. They've all mutated away from functionality in different ways. So all 20 might not work, but if you look at all 20 sequences, you can go back and kind of figure out what their functional ancestor looked like. And we have done that in the laboratory with a structural protein sequence called GAG, and very recently with the protein on the outside of the virus that interacts with the host cell to initiate infection. A lab, like 2 days ago, published their findings in Nature. And the thing is, when we do this, we aren't actually bringing back the entire retrovirus. We are just looking at particular genes, and then we mash them together with a retrovirus that we work with a lot, MLV. And the way we mash them together, it's not functional. Like it can get into cells, but it can't get out again. It's a one-round replication. So what's cool about that is that we can study these proteins and study how they interact with our immune system and how our immune system evolved to defeat these guys. Because all these endogenous retroviruses, they're extinct, especially in humans. There is no modern counterpart to these endogenous retroviruses. So if we figure out how evolution solved these problems, we might figure out how to deal with like HIV or HTLV or the modern retroviruses that cause humans trouble.
Right. So that's what I was going to ask there. So the older ones that somehow our immune system or our genetic machinery has found a way to block them, and so they could no longer infect, they could no longer multiply, and so they've gone extinct. And similar to other life forms, not that we consider viruses a life form, I guess that's debatable in some circles. That the viruses, maybe that their offspring, that they are the ancestors, the ones that are extinct now, that we're finding the fossil record in our genome.
Yeah,
the
like
ones
those
that are existing now.
What I think is cool is that, our endogenous retroviruses, they're extinct. They don't exist in the outside world anymore. However, they've infected every single person on the planet. They're a part of all of our genomes. So what really was evolutionary successful?
Right. You'd think that's pretty successful.
Yeah.
it's there.
It's got every single one of us.
Yeah.
So
yeah, in terms of, at some point I do want to get into the modern retroviruses and the health impacts and what you're looking at there. But in terms of retroviruses, there's such interesting theoretical perspectives on them, especially in this evolutionary viewpoint of, we look at what they do now and they're considered junk. They sit in the genome, they're wrapped up tight, they don't do anything that we know of necessarily or that we found yet. Some people have suggested that retroviruses may have been part of our cognitive expansion, that might have been part of what pushed our human ancestors, however millennia ago, to to having bigger brains, to getting higher thought, that these retroviruses might have been essential to that. Do you have any thoughts on that?
Sure. There are definitely endogenous retroviral proteins that we have co-opted evolutionarily for other purposes. They no longer form a viral function. They form, a human or developmental function. Unquestionably, we have demonstrated that. And sometimes it's the actual proteins from the retroviruses, and sometimes it's just the promoter, the little flag that looks at the transcription machinery and says, hey, make this gene right here. Because retroviruses have really strong promoters. And so if one plops down in front of a gene, then it might be beneficial to make more of that protein. Evolutionarily, we could either wait until, just by chance, we happen to create a promoter that operates, is more functional, or our genome could just steal the promoter from the retrovirus sitting right next to it. And so we definitely have examples of that. However, currently, the way we look at endogenous retroviral protein expression, it might be more of a disease marker because your genome has spent so much time and energy to keep these guys quiet. When they start being able to make a protein, or a protein is expressed on the surface of a cell, or you start getting antibodies to that protein, scientists have started to connect these two diseases, and we might be able to figure out, diagnostic markers, or know a disease might be coming 20 or 30 years before we actually see the effects of that disease.
Wow.
We've seen that with multiple sclerosis, potentially breast cancer, some kinds of lymphoma. So in the modern world, I would say it's more, we currently kind of look at that expression as a disease marker, but there's no question that endogenous retroviruses have played a role in our evolutionary history.
Yeah.
Directly.
Yeah. The chat room has a couple of times come up with the they're excited about the idea of zombie viruses.
Yes.
Is that a possibility? The awakening dead in our genome.
Well, a lot of our retroviruses, endogenous retroviruses, are so old. I mean, I'm trying to think of how old the youngest ones are. They're at least like 30,000 years old. So we can't directly awaken those, but like I said, if we can look at these retroviral families and say this one kind of has a functional this gene and this one kind of has a functional that gene, and kind of piecemeal it together, it would be more of like a Frankenstein virus as opposed to a zombie virus. So you take bits and pieces, put it together, and bring it back to life, as opposed to just bringing it back to life wholesale.
Right. Either way, it's good. Either way, it's very Halloween themed.
Yes, definitely. Some animals, though, do have endogenous retroviruses that are very young, or so young that they still have an exogenous component. And these endogenous retroviruses can still code for functional proteins and functional viruses. It's one of the things we worry about with transplants from other animals. So pigs are actually a really great organ transplant animal, I guess, for humans. But the problem is pigs have a whole bunch of PERVs, porcine endogenous retroviruses. And people have been concerned that if you take a pig liver and give it to a human, then we can have a pig retrovirus in humans, can be transmitted to other humans, etc. So we are worried about zombie ERVs, but not necessarily from humans. From humans, we're going to get the Frankenstein ERVs.
I mean, either way, it's interesting. I mean, just to even know that we understand this much, that we are at a point where we're looking at this level of information is just mind-boggling. Okay,
well,
so
and like with the pigs, it turns out that there was a colony of pigs that were dropped off in some island around Australia that was supposed to be for like shipwrecked sailors. If they happened to wreck on this island, then there would be livestock for them to eat. And it turns out that one of these colonies of pigs didn't have as many endogenous retroviruses as other pigs. And so scientists have taken these pigs and crossbreeded them to get rid of as many endogenous retroviruses as they can to make like a better organ stock for humans. It's crazy what we can do these days.
Yeah, have you gotten? I mean, to take it kind of in the Halloween theme. We're a couple of days before Halloween here. There are some horror novels that kind of start with the idea of these, and in some movies also that start with the idea of these retroviruses, and also retroviruses in other animals like pigs or in cattle, or how you could go on forever. Have you read any of them? Do you think there's any, I mean, if people are asking questions and have this concern, I mean, scientists have a basic concern, but often these concerns get magnified to the nth degree in the popular culture. So do you have a perspective on that?
Well, on the bright side, retroviruses have been with us for millions and millions and millions of years, and we are still here today. And so I think the only problem we need to worry about is maybe like an acutely lethal retrovirus. Like one of the good things about retroviruses is that you might become infected, but the effects of that infection aren't seen for years and years and years. In the case of humans, for decades even. Even in mice, we study a mouse mammary tumor virus in our lab. And in that case, you have to breed these mice over and over and over again before they actually get the mouse mammary tumors. So this isn't the case of like, a crazy outbreak Ebola where, everybody's getting sick and dying, 12 hours later. There is one acutely lethal retrovirus. It was found in a primate colony where they were studying SIV and HIV. It was this acutely lethal SIV, the simian immunodeficiency virus. I'm trying to think of the name, PBJ, SIV-PBJ. And I remember laughing at that because I hear PBJ and I think peanut butter and jelly.
And jelly, right. Yeah.
But it's like this retrovirus that kills you in 24 hours.
Don't eat the sandwich.
Yes. Step away from the peanut butter and jelly. So that would be something scary we should worry about. But as far as retroviruses go, we have drugs that treat most kinds of retroviruses at this point because of the HIV epidemic and because, we keep having to develop new, different antiretrovirals to help those people escape drug resistance. We've got a huge collection of antiretrovirals at our disposal should something happen. I would also say, too, that we've been living with other organisms for millions of years. And what has happened, and we still see happening today in people who hunt primates is for meat, just like, someone in the U.S. might hunt a turkey or a deer. Those actual hunters, if you look in their bloodstream or post-mortem look in their brains, you can find retroviruses from the primates they're hunting. And so that might one day turn into another HIV scenario. However, we're pretty lucky that we've only had one HIV scenario, in the modern world. So it's not something that would be high up on my scary list.
Right. Because like you've said, it's a very rare event that these crossovers do happen, that they do actually end up, probably even rarer, that they end up being something like the really lethal retrovirus.
That was the one. Yeah, that was the only one we have ever found.
Yeah. In terms of modern retroviruses that we're looking at, that you're looking at, what are the big ones? What are retroviruses that people might be familiar with?
Okay, well, definitely human pathogens would be human immunodeficiency virus, HIV, and human T-cell lymphotropic virus, HTLV. And so HIV, of course, many people know causes an immunodeficiency that leaves you susceptible to other infections. And then HTLV causes lymphomas, leukemias, that sort of thing. So again, an immunodeficiency. Ones you might not have heard about are ones like mouse memory tumor virus. Some labs think that it can infect humans and might play a role in some kinds of breast cancer. Another one some of you might not have heard of is XMRV. You might remember I said MLV a little earlier. It's one of our lab strain stock workhorse kind of virus. We actually used it in those gene therapy trials that happened at the turn of the century.
Right.
Accidentally
and somebody died. Did somebody die from that? Yeah.
Someone died from that. But the thing is, the other kids that were treated, they too got this cancer. But when that cancer was treated, the kids were subsequently cured of those diseases. So now that we know to look for that cancer, that has really moved gene therapy forward. But that's a different topic we can talk about in just a second. But XMRV is a virus that some labs have connected with prostate cancer and chronic fatigue syndrome. Man, if you want to start a fight, just mention, XMRV and its connection to prostate cancer or chronic fatigue syndrome. Because, I've dubbed it the magic eye virus, because some people can find it if they look at their data just a certain way, but everybody else is like, what are you looking at? Like, you remember those magic eye pictures?
Right.
Like when you were younger, and if you stared at it long enough, you could see this really cool picture. And there are some labs who are like, whoa, this is so awesome. And everybody else is like, what are you talking about?
I don't really see what you're seeing. Yeah.
Yeah. It's been hard to replicate those findings, and there are many people working on it right now trying to figure it out.
In terms of the findings, in terms of the retroviruses now, the usefulness of them is that they can be used because of the mechanisms they use to get into cells And there are all sorts of medical and therapeutic purposes, like you talked about the therapy at the turn of the century that, which is so weird to say, it sounds so ancient, but anyway, that was 10 years ago, that, went awry then, but there are uses for the retroviruses, right? And they're beneficial.
Yes. And that's kind of what I like to try to talk about on ERV, because people are used to thinking about viruses as the things that make them sick and, things we need to get rid of. But the fact of the matter is, like we talked about before, humans wouldn't exist without endogenous retroviruses. Like, they were an integral part of our evolution. And the thing is, like, we can harness these viruses to do good things for us. So in some cases of, genetic diseases, these kids' lives might be terrible just because they have one gene that is not functioning the way it should be. Well, retroviruses permanently integrate themselves into our genome forever and ever. So why not give a retrovirus a functional copy of that gene that kid needs? Infect him with this, we can make retroviruses harmless in that they can get in, but they can't get out again.
Right.
I kind of mentioned that before. Deliver that in, and then abracadabra, that kid has a functional gene. And that's precisely what happened with kids who had an immunodeficiency. Arnie's starting to play. He's getting a little antsy. But that's exactly what happened. The problem with those initial gene therapy trials is, like I said, the effects of retroviral infection are a lot of times cancer. That's not because the retrovirus carries like an oncogene or it wants to cause cancer. It's evolutionary beneficial to cause cancer. It's a side effect of the fact that retroviruses randomly insert in your genome. And just by chance, it's either going to plop down in front of a native oncogene or in the middle of a tumor suppressor. And when you have billions and billions of viruses going in, it's inevitable that one of them is going to do that.
Right.
And that's
what happens. End up in one of those locations. It's like kind of the luck of the draw. Yeah. But
if we know to look for that now, and we can start these kids on chemotherapy if they show any indication of having cancer, then, they cured, I think it was like 9 out of 10 kids. That's fantastic.
Wow. And
considering the other treatment for this particular disease is the bone marrow transplant, is something like 70% of the kids died. So it's definitely a move or a step in the right direction. And it's not only retroviruses we can do this with. There was just a trial in France where they cured some kids of blindness. Like, the video was insane. So these kids, again, just had one defective gene, and it prevented their retinas from going back to like normal, like an overexposed piece of film. So they used a virus to deliver a gene that can kind of break down the film and make it fresh and new again. And they had this video of this little kid going through an obstacle course that wasn't really an obstacle course. It was just like a piece of paper hanging from the ceiling that he had to step under. And this kid, he couldn't see it all, so he didn't even want to take like one step. After this trial, he was like jumping around and acting like a goofball and zipping all over the place like a normal, 7-year-old kid would act. Awesome. All because of a virus.
Wow. So it's examples like that really, drive home the utility of these things. Someone in the chat room was asking, what is the benefit if you're using it for therapy? How do you, and it's random insertion, do you get away? If it's random insertion, how can it be beneficial? So I'm wondering if in therapy, is it still random insertion, or is it targeted insertion?
In the case of retroviruses, yes, it's still random. And one of the problems is, like we said, epigenetics wants to shut these guys down. Like, it recognizes this doesn't really look like me, rep the DNA. And so a problem with gene therapy is, you might deliver a billion viruses, but, a billion minus one are epigenetically silenced and never actually expressed to a therapeutic effect.
Right.
And so one of the hurdles has been to figure out, what kind of promoter do we need to put on these guys? What kind of cell types are the most susceptible, that sort of thing. With the vision trial, that was actually with an adenovirus. And those viruses are really weird in that they don't really want to insert into your genome, but they will on accident. And when they do insert on accident, it's always in the exact same location. It's a very weird virus and a very weird effect that we're capitalizing on.
Yeah, so they don't want to, but when they do, it's always in the same spot, which is great.
Yeah, yeah.
Great,
yeah.
we know where you're going to go. It's hard to get you to go there, but we're going to make you go there.
Exactly.
In terms of the viruses that that you're working on, I just had a great question and I completely lost it. It just went completely out of my head. Shoop, goodbye. With the retroviruses, here it is. The retroviruses, they're still a virus. So even though we're using it therapeutically, how do you counteract or get around the body's immune system? Does the body still have immune responses that are targeted at retroviruses that evolutionarily we've met in the past in one form or another?
That's a very good question. And that with gene therapy, especially like the adenovirus trials I was talking about, an adenovirus just causes like a common cold. You wouldn't really be able to differentiate that from just a normal cold. And so when you go to do gene therapy with this particular virus, you have to check and see, has this person already been exposed to this virus as an exogenous element? Because if you do, then you're just going to make antibodies to it and it's not going to do anything. And so we've actually played some evolutionary tricks to figure out, can we mix and match these viruses together to make one that your body hasn't seen before, even though it has seen ABCD. Has it seen them all mushed together like that before? In the case of the eye trial, your eyes are kind of immune privileged, so they could get away with putting a virus there and not having an immune response. But what's interesting is that a lot of your endogenous retroviruses, they're self. You don't make immune responses to those viruses. So that has an effect with other animals where they have endogenous retroviruses that still look like exogenous retroviruses. They're more susceptible to infection because, an arm of their immune system has been cut off. But that is a problem with gene therapy with retroviruses in that we can kind of get around it in that, these aren't replicating viruses that you just have to get them in. And if they can get in before your immune system kicks up, then they can deliver and perform their function. Right.
Somebody was just asking again, specifically how the cancer doesn't, how these insertions and changes don't cause cancer. So I think maybe we need to explain that a little bit more clearly. If
they did cause cancer.
Okay.
They did. The adenovirus one, again, with the eye, didn't because of the insertion effect of that particular kind of virus, again, which is not a retrovirus. But when we did do gene therapy with retroviruses, it did cause cancer. But if you treat that cancer and you kill all of the variants that had the insertion that caused the cancer, but you left the other ones alone, then you still got recovery. You still got a therapeutic effect.
But the cancer is not caused in all of the cases, right?
No,
it's caused,
all of the insertions.
Right, in all of the insertions. So cancer is only caused when the insertion ends up inside of a tumor suppressor region, or in an area that either controls a cancer gene or that is a cancer gene that would be expressed or stops the suppression or causes the expression of a cancer gene.
So
right?
let's pretend we only have 10 cells, make this really easy. And 9 of those were infected by a retrovirus and inserted in a totally harmless location. One of them inserted just upstream of, P53 or, one of these cancer genes. Mm-hmm. And that 1 out of 10 caused cancer in a kid and just caused all this trouble. If you can treat the cancer, you can kill those rapidly dividing cells, then those other 9 can go on and populate that body and correct the immunodeficiency.
That's got to be an interesting balance to play between, because you want the cells that are inserted in the way that you want to multiply and to have to create the proteins or whatever it is that you're inserting and trying to cause the cells to do. And so at the same time, you're trying to stop cell multiplication, replication. And so there's got to be this kind of very, very special balance that you have to play between killing cells and keeping them alive at the same time.
Definitely. But it worked. It worked. Whatever they did in that particular case, in that particular protocol worked. And it was kind of a shame that, we totally put on the brakes on that kind of therapy. Because we were worried it wasn't curable, and it wasn't those other kids were either going to lose all of the benefit of the therapy or were going to die too. But they didn't. So I don't know. We should definitely keep moving forward. And I don't think we are in the U.S. as much as they are in Europe. The French, I love them. They're just going nuts with gene therapy with viruses, and they're having fantastic success with it.
Oh, that's amazing. That's good. That's good to hear. I've only heard on the French side of things so far, just the creating viruses in the laboratory based on,
They're crazy. Yeah. Crazy over there.
They do crazy things. But that's great. There's always got to be someone to push the envelope.
Definitely.
Yeah. How did you get interested in all this stuff?
That's a fun story because I was a hardcore pre-med in undergrad. I wanted to go to med school, actually wanted to be a psychiatrist or a surgeon. I was totally into it. And one time we had a HIV researcher come to our university to give a seminar on his research. And I went to a small liberal arts university. We had research, but everything was corn-based. We didn't do any research with viruses or anything.
Right. Corn viruses. No.
Well, funny enough, my undergrad research was with transposons in corn, and they have so much in common with endogenous retroviruses and retroviruses that it's kind of funny that I'm still doing the same kind of research I was when I was a freshman, but totally on accident. Right. But what happened was I was totally enthralled with this fellow's research, and he was like, we should set up this summer research program, between your university and our university, because y'all don't have these kinds of opportunities. And I was like, in my head, I was like, oh my God, that would look so awesome on med school application.
Right.
Doing HIV research. And so I got this internship and I went to do research. And I was like, oh my God, this is so much more fun than the stuff I've been doing volunteering in hospitals and shadowing physicians. Fell madly in love with it. And then I graduated college and I didn't know how to get into grad school because I was so focused on getting into med school. I didn't know what
do I do now? Right.
Yeah.
Right. What's the next step?
So I called that researcher I work for and I was like, do you know anybody who needs a technician or something? Because I graduated and I need work and I don't know what to do. And it turns out that a researcher down the hall from him needed a technician. Nice. And I wanted to find out whether research was something I really wanted to do or whether it was just sort of the summer fun fling thing.
My summer fling with research.
Yes, with HIV research. So I was a technician for a couple of years and, I love it. So I decided to go to grad school.
Awesome. And ever since it's been a love affair with retroviruses and
it has been. I absolutely love it. Any researcher, it's like what we fall in love with, we will talk about ad nauseum until our audience's eyes just, glaze over and people start passing out from boredom. And we're all that way.
No.
Yeah. Okay, I'm totally that way.
Exactly. And for your research that you're working on, let's talk a little bit about you're not doing are you doing animal studies or are you just doing virus basic research?
Yeah, is tissue culture or just straight genetics based. We do have animal work going on in our lab, but it's not what I do. Okay.
And your focus in human health, how has that grown? Talking about HIV, it's something that has been a problem, has been a growing problem around the world for years now.
Yeah. Well, endogenous retroviruses have definitely played a role in, how I describe evolution to the general public, because a lot of the times the general public just looks at evolution as, a dinosaur evolving into a bird, evolving into a hamster, evolving into a monkey, evolving to a human. Like, it's something that happened millions and millions of years ago that they don't really have a connection to. But if I can talk about endogenous retroviruses and how, these bits of evolution connect to their aunt's MS, all of a sudden, evolution and retroviruses are something they can care about and connect with personally. Yeah. And so I found that's been really effective, talking about evolution with the public. As far as HIV goes, my summer research project was studying HIV-infected infants.
Oh, wow.
And, I mean, come on. It's as heartbreaking as anything on this planet can be. And, half of my cohort was kids who were dead by 3 to 6 months of age. So it's definitely been a motivator. I mean, I don't know. It's hard not to be motivated in this particular field. And it's hard not to be discouraged a lot of the times too, because, we literally have the best minds of our generation trying to solve this problem. And we're trying so hard. And sometimes it just feels like we're treading water. Like we're keeping up with the virus, but how long can we keep treading water?
Right.
At the same time, it's also really exciting because we've got so much awesome technology at our disposal today that we didn't have in 1980 when this epidemic came up.
Yep.
So the way we can study the virus now, and especially, the way I personally am studying it in the lab, I'm very optimistic that we can figure this out. And not only in a treatment fashion, but in a vaccine, that, we can nip this in the bud. I think we can do it.
Wow. So you think that, do you think it would be better, instead of continuing forward, or maybe in addition to continuing forward with antiretrovirals, that a vaccine is actually possible for HIV?
Yes. And I think we have to make it possible because, we're in this continual race with bacteria and trying to create antibiotics that, we can use to treat these bacteria. And they're almost, figuring them out before we do.
Yeah.
And we do not want to get caught in that race with HIV. We're currently staying ahead of it. But again, there's only so many options, and there's no way out of that rat race, the evolutionary rat race, because, if HIV can sample the sequence space of, billions and billions and billions of sequences in one patient over the course of their lifetime, we aren't going to win that race at all. So the only way to stop HIV is to stop that initial infection in the first place. The only way you can do that is with a vaccine or condoms.
Right. Yeah, or condoms,
For real.
Or abstinence, but that's probably not going to happen. With the vaccine, the challenge though is the fact that there are multiple strains of HIV, and the fact that it changes.
Definitely.
So that makes it in a sense. I mean, I look at like the flu virus. The flu virus is not a retrovirus, but at the same time, every year it's constantly recombining, and every year there needs to be a new vaccine, and they're searching for a universal vaccine, one that takes some kind of common component that it's always the same that we can vaccinate for. Is that similar? Well, do you think there is a universal vaccine for HIV?
We have figured out some of the chinks in HIV's armor. We might not have to vaccinate against every single variant, because it turns out that, how some people you've heard, progress to AIDS from HIV infection very quickly, and some people progress slowly.
Right.
Certainly a lot of that has to do with that person's immune system and, their stressors and that sort of thing. But a large component of that is the general evolutionary fitness of the virus they're initially infected with. So someone who is progressing very quickly, if you take viruses out of them and compete them like in a head-to-head competition with viruses isolated from someone who's progressing slowly, the viruses from the fast progressor always win. And some labs have found that, an HIV transmission, there's only like one or two viruses that establish that infection. And some labs think that the ones that get through are also more evolutionary fit than the ones that don't get through. So we don't necessarily have to vaccinate against every variant. We need to figure out what the more fit variants look like and vaccinate against those.
Right.
Because you can either prevent infection directly, or someone will be infected with a virus that is fundamentally less fit, and they'll be able to live longer, hopefully without starting antiretrovirals, hopefully not getting into that, continual race between the antiretrovirals and the virus itself. So there's little hints that HIV has dropped like that one that are leading us towards more logical vaccine design. The problem with current HIV vaccines is that we don't really know what to put in them. So we're just putting in like a lab strain. We have no idea whether that's a good idea or not, but it's just a variant we have. So we're like, let's go with it. So a huge step in the right direction is figuring out what is the right thing to put in a vaccine. And the Los Alamos sequence database, they've collected every single HIV sequence ever sequenced. And they figured out what the most common, kind of the universal HIV sequence. And they're going to start putting that into a vaccine to see if, if this particular sequence of amino acids is usually there, why not put that in the vaccine and see if that actually helps people? It's a better idea than what we've been doing. And I think we're going to continue to get more better ideas.
Yeah, that sounds like a real step in the right direction. I love the bringing together the modern technology, gene sequencing and genome sequencing, and the future of vaccines. And hopefully it would be great if we could get a vaccine. That would be amazing. We are running out of time here. I just wanted to ask a final question just about or get into a final couple of questions about your blog. You do blog at Science Blogs /ERV, ERV. What started you blogging?
I moved to Oklahoma. Yeah. And I just moved here when my boss got a position at the university, and I didn't know anyone here. I didn't know really what the local science groups are about. And so I really kind of felt isolated. So I started screaming into the dark on a Blogspot blog. I
hear this story. It's a common thread among a lot of bloggers, is this start of, I don't know if you know Vincent Racaniello. He does
yes.
This Week in Virology. He has said something very similar about starting his blog. It was like, I just started writing, just put it out there, don't know if anybody's reading it. And nobody did read it at first. I mean, you set up like SiteMeter and there's 0 and you're like, awesome, I'm gonna keep writing this anyway. And you get readers by just leaving comments on other people's blogs and being different. I mean, I didn't know of any other blogs out there that focused on viruses when I first started, and I was like, there's a niche here, I can fill it. And a lot of it was just I needed a place to vent about, things I'd read in the news, or, stupid things politicians were doing, or just like cool things that I learned in Journal Club or something. I needed it more than I needed people to read it. And then people started reading it, and I was like, awesome. You're like, yay, I'll keep going with this.
Yeah, why not?
what kind of stuff do you tend to focus on in your blog? There's the viruses, there's retroviruses, but you also you expand on that a little bit more.
Yeah. A lot of the times I'm like the Debbie Downer and Negative Nancy on research. Yes. Someone will talk about this really awesome new finding either in HIV or in virology. And then I go read the paper, I'm all excited, and it's like, ugh, this isn't as cool as the press release made it out to be. And so sometimes I have to be the Debbie Downer. Sometimes I get to point out really cool research that isn't getting a lot of press that I think should be. Talk a lot about anti-vaccination efforts, of course. I love my viruses, but I love polio being extinct. That's not in smallpox. That's not a problem.
Right.
And then let's see, HIV denial. That is frustrating, but at the same time, it's also kind of entertaining because just the personalities involved in that particular movement they're so kooky and so off the wall that at least with my audience, we can have fun with it and appreciate the kookiness as opposed to getting focused on how awful the things they're saying are and, the effects of what they're saying. Let's see, I write a lot about just general skepticism, science outreach, that sort of thing. There we are live.
Yes.
Let's see.
Bringing up your blog right now so people can take a look at it. Yeah. So just within the first page, you have, hey, look, it's us on the Science Hour, and something about an
Did you hear that?
Yes, your dog is going insane.
He's throwing a fit.
Tantrum time. Don't worry, we're almost done. It's really funny. Another one. Rebutting somebody online.
Well, no, what happened in that case, this is a stem cell controversy I just read about in Nature, where this person, they say they're a group, but I'm certain they are a person. They're emailing journalists and universities and colleagues of individuals in journals saying that, scientist XYZ has committed fraud. And then they back up that accusation with comments like image 4A looks weird.
And specific.
Yeah. And then they point out things like, they used the same cells in image 4A, B, and C. When you read that paper, yes, they did. They were showing that those same cells expressed protein A, B, and C. They were showing fluorescence and co-localization of that fluorescence, and et cetera. This person is either an undergrad or not in the field and doesn't really understand the papers they're reading that just happen to be on embryonic cell research and accusing everybody of fraud. And as a scientist, accusing a scientist of fraud is the worst
business. Yeah.
You can accuse me of anything and I don't care, but accuse me of fraud, I will freak out. Because, scientists, that's what we have is our trust in one another. That, I'm honestly collecting this data and honestly presenting it to the public to be assessed and replicated and to move the field forward. And this person is just willy-nilly accusing people of fraud and the effects of that anonymously. It's irresponsible and it horrifies me. So I thought it
shouldn't be allowed to just continue without anybody saying anything about it. That it should be brought out into the open and there should be people talking about it. So
sure. And if you genuinely think that someone perpetuated fraud, it's like you need to get the evidence to support that assertion. You need to go through the proper channels. You don't go straight to a journalist and say, this paper in PNAS or Nature was fraudulent. There's a way you go about correcting these errors, and that's not the way this person's doing it.
Well, I'm glad there are people like you out there who are bringing things like this to light, who are talking about science, who are, I mean, the function of your blog that, started out as a very, just individual attempt. Now you're out there educating people, responding to the press release and saying, well, it's not exactly right, getting accuracy out there and promoting science. So you play a very important role in the entire science communication ecosystem, and in addition to doing some pretty awesome research. So thank you so much for joining me today.
No problem. I had a ton of fun. Be happy to come back.
Awesome.
Probably not during Arnie's walkie time, though. He is just like stalking behind me. I can hear his little collar going tink, 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 tink.
He's like, come on, time to go for a walk.
Yep. Yeah.
Well, we have reached the end of the hour. So once again, thank you very much. I've had a lot of fun talking to you too. This is just this topic is just so interesting, and it deserves more time. And I'm sure that we will have a chance to get to that at a later date. But in the meantime, everyone out there, if you want more information, if you want to keep up with the writings and the scientific life of Abbie Smith as she gets her graduate degree, you can go to scienceblogs.com/ERV. And additionally, you can check out more Science Hour stuff. Abbie, are you on Twitter?
I am not on Twitter. Everybody else is on Twitter. I'm not on Twitter. Sorry.
All right. Well, I can't tell people to go find you on Twitter. Go to your site. Go to her science blog. Go to her blog. Exactly. Everyone out there, thank you so much for watching. Do appreciate this hour out of your day. If you are just catching this, want to catch the episode, watch it later. You can watch this episode and past episodes at twit.tv/Kiki. There are all sorts of episodes there. And hey, we're also on YouTube, the whole Twit account on YouTube. You can find all this stuff, embeddable video. It's all really exciting. You can also follow other sciencey pursuits of mine online. Just follow me on Twitter, Dr. Kiki. And additionally, I have This Week in Science, TWIS, every Thursday night, live here on the Twit Network at 7:30 p.m. Pacific Time. Dr. Kiki Science Hour is every Thursday, 4 to 5 p.m. Pacific Time. And also Green Tech Today, our new Green Tech program. We're highlighting our choices for the top 25 green tech innovators. That's on Mondays, like 1:30 p.m., I do believe, Pacific Time. I think that's about it. If you want, you can come back next week. I'm going to be here. We're going to be talking about materials. What's interesting about materials? You'll find out. There's lots of materials out there for us to talk about related to hippo sweat and spider silk. Not necessarily all at the same time, though. Come back next week. It's just an hour, and that's all I ask to make your world a whole lot more interesting. Thanks a lot.